Hi everyone, what is up? Am I audible? Hello? Hi everyone, am I audible? Up. Hi everyone, what is up? Am I audible? Hello? Guys, okay, let me adjust the volume. Okay, so now am I perfectly audible? Hi everyone, now am I perfectly audible? Yes, do let me know in the chat section. Okay guys, so today we are going to start respiration in plants. Okay, so according to me this is a very easy chapter and an important one, but I don't know why students find it very difficult, so I'll try my best to make it very easy for you. Secondly, students, you know that we are already done with the breathing and exchange of gases. Now this is the chapter from the plant physiology. So I was thinking, rather than completing human physiology first then starting plant physiology, it's better to pick up one chapter from this, one chapter from that. Is that okay for all of you? Is that okay for all of you? I believe you all are fine with it. So let's start the chapter and make sure you have your NCERT with you, right? Make sure you carry the notes today. So whatever I'm going to mark, right? Whatever I'm going to highlight, please make sure that you do the same in your book. And other than that, right? In this chapter, I'll provide you my handwritten notes as well. And that is it. Like, this is what you need. Nothing else. Nothing else. Everything from this chapter will be completed. So let's get started. So the name of the chapter is the Respiration in Plants. What is the name of the chapter, students? The name of the chapter is the Respiration in Plants. So first of all, before proceeding it, do let me know what is the meaning of respiration. Yes, everyone. In the chat section, I want you people to reply this, to answer this. So quickly tell me, what do you understand with the word respiration? You all are well aware what breathing is, right? In the last class also told you about the respiration. Now you have to tell me. I'll see whether you are revising or not. Quick guys. Quick everyone. So let's talk about the respiration, that what exactly is the respiration. Quick. Do you know about respiration? So basically we know that, first of all, if I talk about the humans, you know it very well that the very first step of the respiration is the breathing. That is pulmonary ventilation. That is, we inhale the air, right? We inhale the air and obviously we exhale the CO2. In that air, we know that mixture of gases it is, right? We talk about the partial pressure of oxygen and everything. So in the last lecture also, I told you this thing, that oxygen is required for the oxidation of food. Right, but the very first step, as I said, is what? The very first step is basically the breathing. So when you are talking about the respiration, ultimately what are you talking? You are talking about the breaking of carbon-carbon bond. That's what we are discussing here. Right, we are talking about the carbon-carbon bond, the breaking of this carbon-carbon bond, which is present in the sugar, and to be more precise, which is present in the glucose. So we break that bond, right? That glucose will be oxidized by using oxygen, and that will be done in multiple steps by using enzymes, so that we can trap all the energy in the form of ATPs. Are you getting my point? Yes. Are you getting my point? So that is what we are discussing here in this particular chapter. So what are the main and the important topics of this particular chapter? So here in this particular chapter, you will talk about the, I'm picking up the main points. You will talk about the glycolysis. We will also talk about the fermentation, guys. We are also going to talk about the Krebs cycle, and then you will talk about the ETC, electron transport chain, and about the RQ value. So these are the most important topics that we are going to discuss here. So you tell me which one is your not so favorite. Quick. Which one is not your not so favorite? Quickly tell me. And you know, yesterday I received these books from Unacademy. Do you know about them? right? So there's like I received all the notes, class 11, 12, even the flashcards were there, and you know the remaining chapters, they were in the separate book. So very quick revision, like that will be very helpful for the quick revision. I got this, right? QRTS, right? Your basically quick revision. Even I got your URTS, that is Unacademy revision test series as well. I was just checking out the paper. So soon, you know, even I will sit, I'll pick up each and every paper, I'll sit and I'll see how much time do I take to complete the biology part. We'll surely discuss that as well. Okay, so I got one glycolysis part. So you find it difficult? Arey yaar, glycolysis is the easiest topic here. Anything else? Arey yaar, everything is easy. Everything is easy. So let's get started. Okay, let's get started. So these are the topics that we have to study. So as I said, the very first thing is respiration. Yes, everyone, the very first thing that we have is the respiration. So I am writing the definition here, right? I am writing the definition here, and I have already explained you this. So I hope you all will write down. So basically you are talking about the, right? You are talking about the, yes, please, cellular respiration first, right? Cellular respiration first. Okay, and here if you think that it is the respiration in plants, so only plants will show that, no, you'll see this in our body also. So cellular respiration is a process in which there is a breaking of, in which there is a breaking of carbon-carbon bond through, how? Through oxidation. There is a breaking of carbon-carbon bond, right? It's a process in which there is a breaking of carbon-carbon bond through oxidation, right? In an enzymatically controlled multi-step process, right? Multi-step process. Noted? Yes, everyone. Noted. So this is what cellular respiration is. Is that clear? This is what cellular respiration is. So it is a process in which there is the breaking of carbon-carbon bond through oxidation in an enzymatically controlled multi-step process. So each and every word written here is very, very, very important. So this is what you have to keep in your mind. Am I clear, guys? Am I clear? Now my question is that why do we have to do it in multiple steps? Why can't we can do it in one step only? Yes, please. Yes, please. Tell me quickly. Why? Why is it so? Why are we going for the multiple steps, right? Not for the single step? Because if breakdown is there in single step, if breakdown is there in single step, then heat will be produced, heat will be released, guys, and here we have to trap this energy, right, in the form of ATPs, right? We know that ATP is our energy currency. So here, if we will go for one step, then what is going to happen, guys? If we will go for the one step only, then let me tell you what this thing will happen, that here the ATP, the heat will be produced. Okay, okay, right? So then it will be converted into the heat. So that is why we are not going for that. Okay, that is why we are not going for that. So here, as I said, so this will be done so that we can form what? We can form the ATP. Okay, so cellular respiration process in which there is breaking of carbon-carbon bond through oxidation in an enzymatically controlled multi-step process to form ATP. So we know that from class 9th we are reading this ATP, that is adenosine triphosphate. That is what? Adenosine triphosphate. We know that it is the energy currency of our cell. So whenever cell is in need, right, it is the energy currency of cell. So whenever cell is in need, these ATPs will be used. I hope this part is clear to everybody. Understood? Understood? Right? So now what is the next step? Next step is the compounds that are oxidized. Compounds that are oxidized during respiration. What do we call them? are respiratory substrates. What do you call it, students? You are going to call it as respiratory substrates. Am I clear? Yes, everyone. Am I clear? Sure. So these are the respiratory substrate. So when you talk about the respiratory substrate, the main one is carbohydrates. So I really want to know why. Yes, I really want to know why. I'm checking out the chat so that you can revert quick. Right. So these are the basics. And if your basics are clear, then obviously you are not going to find it difficult at all. So tell me. Tell me. Yes, please. So respiratory substrate, we know that usually it is what? Usually carbohydrates. Usually what? Usually carbohydrates, but your fats, even organic acids, etc. can also act as respiratory substrate. Am I clear? They can also act as respiratory substrate, but always remember, when it comes to the main respiratory substrate, right, when it comes to the main respiratory substrate, then it is nothing but the carbohydrates. Okay, you know it very well. So it is what? It is carbohydrates, and you can also call, say here, to be more precise, you can talk about the glucose. Is that clear? Is that clear? So why is it so? Why is it so? So main one is glucose. But if, let's suppose, if it is not available, then obviously body will go for what? Body will go for the fats. Body will go for the proteins then, right? So main respiratory substrate is carbohydrate. So if not available, then fats, then proteins. Okay, so that's what you have to understand. Okay, so then fats, and then what? Then proteins. So this is what we need to keep in our mind. So again, multiple steps, making ATP, using ATP when there is need. Main respiratory substrate is carbohydrate. If not available, body can go for the fats, body can go for the proteins, body can go for the organic acids. Is that clear? Is that clear? So here, before going forward, overall, if I talk about the respiration, okay, overall, I'll talk about their calorific and physiological values as well. So overall, if I talk about the cellular respiration, overall, I'm telling you, read the NCERT, read the last part of this chapter. You will find it is mentioned that cellular respiration is amphibolic process. But what exactly is amphibolic? Do you know that? What exactly is amphibolic? So amphi means dual. We know it very well. So when you are talking about the amphibolic process here, so students, you will see catabolic. Amphibolic means dual process. Like here, catabolic and anabolic processes are occurring simultaneously. So if in general I talk about the cellular respiration, we say it is amphibolic. Read the NCERT, find out the last part. It is clearly mentioned that respiration is considered as amphibolic. So amphibolic means here in the respiration, catabolism. So cata, I told you already, cata means cut, to break. So catabolic processes means where what is there? Where breakdown is there, and here what is there? There is the buildup. There is what? There is buildup. So overall in respiration, breakdown and buildup is there, right? Breakdown and buildup is there. But in general, like see, where many students make mistake, right? Like when ma'am, we talk about the cellular respiration, if we are talking about the breaking of carbon-carbon bond, right? If we talk about the breaking of this carbon-carbon bond, then it should be catabolic. So why are you saying, right? Why are you saying that it is amphibolic? It's simple. With time, obviously, you'll get to know. See, no doubt initially you are just breaking that carbon-carbon bond, but overall in respiration, it is not just the bond breaking that we are doing, right? It is not just the bond breaking that we are doing. Here, bond breaking, making, both the things will be there. Whenever there is a requirement in the body, right, body will convert one particular metabolite into the another as well, right? Accordingly, breakdown, buildup is there. So overall, if I have to see this process, so what will I say? That it is going to be what? It is going to be amphibolic. Am I clear? It is going to be what? It is going to be amphibolic. Now, here we were saying that carbohydrates, right? That carbohydrates are the main respiratory substrate. Then fats, then proteins. But overall, if I have to tell you, students, fats will give you more value, more energy. But still we go for the carbohydrates. What can be the possible reason? It's simple. See, For the carbohydrates, when you break them, when you oxidize them, right? Obviously, when you oxidize them, we in this chapter itself we will discuss. We also talk about the respiratory quotients, right? We see that what is the volume of oxygen used, what is the volume of CO2 evolved. So accordingly, we have the respiratory quotient values as well. Okay. So when it comes to the carbohydrates, we oxidize the carbohydrates, right? In multiple steps, we know it very well. Now, what is going to happen? If in comparison to fats, we know it very well, carbohydrates are easy to oxidize, right? In our body, we need the energy source. We are eating the food. Why? We need the energy, right? We are breaking that complex food into the simplest one, ultimately into the glucose, right? And then we are breaking it, we are oxidizing it so that we can get the energy. So in our body, if I talk about the brain cells also, they need the energy immediately. Like if for few seconds, or I would say for milliseconds even, if your brain will not get the glucose, not get the energy, then what will happen? Stroke can occur, paralysis can occur. So we need the energy source which can give us the instant energy, number one. Secondly, in the case of fats, you know about the fats. You know there are steps involved when it comes to the digestion of the fats. We have to break them in from, we have to break these fats. We have to convert their complex form into the simpler one. You know that, right? Then like emulsification of fat, and we know that water insoluble they are. Then you know the detailed process. I believe from class 10th you remember it a bit. So what I want to say, in the case of fats, if you have to oxidize them, you have to use a lot of oxygen as well. No doubt it will give you more energy, but you have to use a lot of oxygen there also. So that is why we need the energy source which can give us the instant, which is easily oxidizable, can give us the energy instantly, right? And why not proteins? Because in the other things are also there, their byproducts also, but in the proteins also, you know that our body, protoplasm, majorly what is there? Proteins are there, right? Our general framework, enzymes, they are proteins. So if body will start digesting on the proteins, then it will break our basic structural framework also. Okay, so when you are not eating, right? Initially body, what that body is going to do? Initially it will break the carbohydrate, the stored like glycogen and all. Then it will shift to the fats. So if you will see somebody who's on a kind of hunger strike for a long period of time, or whatsoever is the reason, you'll see, right, they'll become very lean or something. Why? Because body, after carbohydrates, body will go for the fats, and then finally body will go for the proteins. So when body start digesting the proteins, we call it as the protoplasmic respiration, and obviously many toxic substances will form in our body. This is not good, and if body has started this, then obviously it is risky. Is that clear? Is that clear? Is that clear? Sure. So that's what you have to remember here, right? That's what you have to remember here. Now, I will add one more concept here, right? I would like to add one more concept here, and here we are going to talk about the calorific value and the physiological value. Okay? So this is our respiratory substrate. Let's talk about the carbs first. Let's talk about the fats, and then we have the proteins. So there is something called as gross calorific value, and there is something which is known as physiological value. Do you know the differences in that? Yes, please. Do you know the differences in these? The units will be kilocalorie per gram here. So do you know the differences there? Anybody? I'll give you a very relatable example. Right? Just say, let's suppose today you just got motivated after watching my motivational video, and now you are like, today I will study. I will give my best. You make a timetable then. You make a timetable then. As per that timetable, you will study for 12 hours a day. How much? For 12 hours a day, I am going to study, right? I will not waste even a single minute, even a single second. This is what you decide. But because in your timetable or in your target table, you are planning 12 hours, but at the end, you just end up in 7 hours. And that too much, right? Mostly, ma'am, we just end up in 5 hours or in 3 or 4 hours. So this 12 hours is your gross calorific value, to be very honest, and the one that you have achieved is your physiological value. Are you getting my point? So the one that you planned, That's how I'm going to explain it to you, right? So that is your gross calorific value, and the one that you have achieved, we will call it as the, yes please, we will call it as the physiological value. Is that clear? Is that clear? Yes please. Done bache? So that's what you need to understand here. Okay, so now in general, so ma'am, is it so that the carbohydrates, what is the like in case of carbohydrate, protein and fats, how is it related? So when it is the gross calorific value, it is the total amount of energy released. What is gross calorific value? You don't need to write the definition, but yeah, you need to understand it, right? Just say, let's suppose you know the physics in the bomb calorimeter. If you go for 1 gram of the respiratory substrate, how much? 1 gram of the respiratory substrate, then the total amount of energy that it is going to release in that bomb calorimeter, the total amount of energy that it is going to release after the complete oxidation will be the gross calorific value. So I will define it again. Gross calorific value means basically it is the amount of energy, total amount of energy, right? It is the total amount of energy that is released by 1 gram of respiratory substrate, right, when it is completely oxidized in the bomb calorimeter. That will be your gross calorific value. But what is the physiological one? The actual one, right? The actual one which will be obtained in the body from that same 1 gram of the respiratory substrate. Right, that's why I related it with the timetable. Got that everybody? Got that everybody? So obviously here what you have to see, you will see, so this is the physiological value. Is that clear? Yes, is that clear? Right, so physiological value, obviously it will always be less than this gross calorific value, because you know that a lot of it will be wasted, right? Right, so I'll give you the example. So in the case of carbohydrate, it is going to be 4.1 kilocalorie per gram. But here there is no major change. Here you will see this is going to be the value. Are you getting my point? This is going to be the value. This is what you have to see. Here it is going to be 9.45 kilocalorie per gram. See, I told you. It is the more energy than the carbohydrates, but still they are not our primary substrate. So it is 9.45 kilocalorie per gram, and here it is going to be 9 kilocalorie per gram. You can see the difference here as well. And for the proteins, you will see that it is going to be around 5.65 around kilocalories per gram, and here it will be 4 kilocalories per gram. So that's what you have to remember, everybody. Am I clear here? Yes, please. So this is the gross calorific value. It is the total amount of energy released by 1 gram of respiratory substrate when it is completely oxidized in a bomb calorimeter, and this one is the actual one that will be obtained in the body. So you can see the difference as well. You can see fats are giving more energy, but still they are not our main respiratory substrate. So in the comment section, I want you people to write more points that why fats are not considered as the primary or main respiratory substrate. Okay, so this is about the basic introduction. And here when you talk about the cellular respiration, just keep it in your mind, it is going to release the energy, right? So what's going to happen here, guys? Energy will release. Okay, energy will release. Any doubt so far? So let's have a 1 minute ka revision quickly. So we started with the respiration. We said that the cellular respiration is a process in which there is the breaking of this carbon-carbon bond, right? And that through oxidation in multiple steps by using enzymes, and ultimately what is formed? ATP is formed. It is the energy currency of our cell. So we know that it will not be done in one step, because otherwise heat will be produced. But here what are we doing? We are breaking them into the multiple steps so that we can trap that energy in the form of that high energy bonds of ATP. Is that clear? Is that clear? So compounds which are oxidized, they are the respiratory substrate. Main is carbohydrate. If not available, body will go for the fats. Even in plants, in some seeds, you will see organic acids are also there. Proteins are there, right? So you know the sequence here. Is that clear? You know the sequence here. Now you know the this kilocalorie thing as well. Am I clear? Am I clear? So after this, let's have a look of this from the NCERT. So these are the basic points, guys. Make sure you underline it as well, right? See, you have to read this book at the end. If somebody says, na, just read my notes, they are the more sufficient, right? They are amazing, they are this, they include all the NCERT. If it is made by you, you can consider that. But still, it is my advice to read this book. Its description and all. If you are reading this book and after that, right, if you are reading this book and then after that you are making handwritten notes, short notes, follow that. Otherwise, we have to read this book, right? The diagrams, the description, the summary part, NCERT exemplar questions as well. Do not forget about the NCERT exemplar questions as well. Understood? So here you people can see all of us breathe to live, but why is breathing so essential, right? Already told you that all living organisms, they need energy for conducting their day-to-day life. So here there is nothing that you have to underline. So we need the energy. How are we getting it? I'll tell you, I'll give you the NCERT highlights as well. So there are many questions, right? But in reality, we know that process of breathing is very much connected to the process of release of energy from the food. All the energy for the life processes, how are we getting it? By oxidation of these macromolecules, which we are calling as food, right? Right, so only green plants and the cyanobacteria. So green plants, you know that photosynthetic even. What is cyanobacteria? Your blue-green algae, right? What is cyanobacteria? Your blue-green algae belongs to Monera. It is what? Photosynthetic. We know that. These are the first organisms on this earth which releases the oxygen, right? They do the oxygenic, they perform the oxygenic photosynthesis, right? So what they do in the photosynthesis, they just trap the light energy, convert it into the chemical energy that will be stored in the bonds of carbohydrate like sucrose, glucose, starch, etc. Is that clear? Is that clear? So in the green part, you know that, right? So green plants we must remember that in green plants too, not all cells, tissue and green photosynthesize. You know that chloroplast, it is that main organelle for that, right? So in green plants, all other organs, tissue and cell that are non-green, they also need the food for oxidation. There also the respiration will occur. So if you think that why in plants respiration is there, they do the photosynthesis. Obviously, non-green parts, what will they do? How will they get the energy? They have to oxidize the food, right? So we can send, like in the leaves, photosynthesis is taking place. So the food, with the help of phloem, it will be translocated to the non-green parts. And for the animals, we know it very well, they are dependent on the autotrophs for their food as well. Okay, so here they have mentioned the cellular respiration, that we will discuss it in this chapter. So not exactly, there is something which you have to underline here if your basics are clear. If not, you find some line to be line that I might forget it later, so you can underline it. Now photosynthesis, we know that where is it taking place? It is taking place within the chloroplast. Can you see that, guys? It is taking place within the chloroplast. So what is happening? There is the breakdown of complex food through yield the energy. Is that clear? Which organelle is involved there? Of course, mitochondria and eukaryotes. But when there is the breaking of carbon-carbon bond of these complex compound through oxidation within the cells, leading to release of considerable amount of energy, this is the respiration, and the compounds which are oxidized are respiratory substrate. Understood? Understood. In the lecture itself, we will clear our NCERT. So usually it is the carbohydrates, right? Told you already. Usually it is the carbohydrates that will be oxidized, but protein, fats, they are also used under certain conditions. So during oxidation within a cell, all the energy contained in respiratory substrate is not released free into the cell, right? It will not release free into the cell. Okay, beta, in a single step. We know that it will occur in slow stepwise reactions controlled by enzymes, and then energy will be trapped in the form of ATP. So all these points, we have just summarized that in one definition. Understood? Understood? Right? So another next important point. So they are saying that the energy released by oxidation in respiration is not used directly, but is used to synthesize ATP. Is that clear? Right? Which is broken down whenever energy needs to be utilized. So this energy, right, it is not used directly, but we know that it is stored in the bonds here. Understood? So ATP is the energy currency. So you can see that in the notes, we are completing our understand our NCERT, and we are covering this NCERT in detail. Okay, okay, chale. So what's the next part? So we will discuss it from here only. I'm not going to write it, right? So do plants breathe? What do you think? Do plants breathe? What do you think, guys? Do plants breathe? Yes. Do plants breathe? Of course, they do. They do indeed. Indeed. Yes, they do breathe, students. They do breathe. You know, our elders, they used to say that we should not sleep under the tree during the night time. Why? Because then the plants, they will not go for the photosynthesis, rather they will go for the respiration. So we know that in the respiration, which metabolic gas will be released? Your carbon dioxide, isn't it? So here you can see plants required oxygen for respiration to occur, but they also give out CO2, right? Right? So plants, but unlike animals, plants, they do not have any specialized organ for that. Like we have the lungs, they do not have anything like that. Why is it so? Because each and every part is capable of fulfilling its own needs. Like if I talk about the stem, right? In the stem, you know that we talk about some pores, lenticels. Do you know that we talk about the lenticels there? Like if you see a woody plant, there will be the bark there, but bark, which is a non-technical term, you see the bark there, but in between you will see that living tissue is also there. They are forming the lenticels through which gaseous exchange can be done. If I talk about the leaf, I talk about the stomata, right? The gaseous exchange is done. Plus, in the case of plants, the requirement is not that much. They are fixed to a place. Their metabolism in comparison to animal is quite slow. Animals, they are moving here and there in search of their food, for mating, for their protection. But in the case of plants, they are at one place only. Their demand is less, right? And each and every part is capable of fulfilling its need, right? So they do not have the specialized organ for this, but they do have stomata. They do have lenticels for this purpose. So yes, you should know about the lenticels, right? You should know about these lenticels, right? Right? So you know that in plants, each plant part, it takes care of its own gaseous exchange needs. Told you already. If you think that question cannot come from this part, it can come from this part. In the statement-based question, they can ask you this. So there is a very little transport of gas from one part to another. What is the second reason here? Plants do not present great demand for the gaseous exchange. I told you why. So roots, stem, leaves, they respire at rates far lower than the animals do. So only during photosynthesis, large volume of gases are exchanged, and each leaf is well adapted to take care of its own needs during this period. Okay, okay. So when cells, they photosynthesize, availability of oxygen is not a problem, since oxygen is released within the cell. And third reason is that the distance that gases must diffuse even in large bulky plant is not that great. Okay, because each living cell, it's quite close to the surface of the plant, right? So they do not have the requirement of, you know, that special respiratory organs. I told you the reasons as well. Okay, now here also they are saying, what about the thick woody stems and roots? So stems mein living cells are organized in thin layers. We used to read it. Now, so it is deleted from your syllabus, but we used to read it in anatomy. Okay, so there are thin layers inside and beneath the bark. So bark, it's a non-technical term. It's not a technical term. It's a non-technical term. Am I clear? Okay, so they have the openings. We are calling them as lenticels, right? If I don't know whether you know it or not. So there used to be a diagram, like let's suppose here you'll see the cell, you'll find this a pore like structure here, right? And here there are many cell layers, right? This diagram used to be there in the NCERT. Okay, okay. Acha ma'am, start morphology of flowering plants. Beta, we will start it soon, but whatever chapters we are discussing, at least complete that first, hai na? Why is it so? Whenever we teach something, like let's suppose if a teacher is teaching X chapter, then some students will come, they'll like, no, start Y. If that teacher will start Y, then students will like, oh, go for this chapter. Why, bhai? Why is it so? Why is it so? Okay, so this part is clear, everybody. This part is clear. So lenticels, so cells in the interior are dead. They are dead and provide only mechanical support. So thus, most of the cells of a plant, they have at least a part of their surface in contact with air. So that's why they don't need the special respiratory organ. Their demand is also less, right? So when it comes to the lenticel, here you will see loosely arranged parenchyma cells, right, which will help. And moreover, even in leaves, even in stems, we have that type of cortex. We have seen that, okay? So this is all about it. So that's what you have to remember. So complete combustion of glucose will produce CO2 and water, and the end product will yield the energy, most of which will be given as heat. So there is a difference in the respiration as well as in combustion. Can you tell me? Yes? Please tell me about that. Quick. Yes? What is the difference in respiration and combustion? Quick. Anybody? Thank you, beta Ravia. Thank you so much, beta. So next is, see, in the case of respiration, that's just a slow process, multi-step process, enzymatically controlled. But if you talk about the combustion, it is rapid, right? It is not a multi-step process. It is the uncontrolled step process, right? So respiration will take place within the living cell, students, mainly mitochondria. But when you talk about this combustion, it will be outside the living cells, right? And for combustion, we have to provide the high temperature. Respiration will normally occur at our body temperature. So these are the things. Baki, you can see that here the heat will be produced, and here we know that in the respiration, energy will be released gradually in steps, and we can trap that. So there are the differences. There are the differences as well. Is that clear? Is that clear? So that's what you have to remember. Now, let's go forward. So, see, when we talk about the evolution, I'll relate it with the evolution now. When we talk about the evolution, right, when we talk about the origin of the cell, right, and all, we say that initial cells are primitive. Firstly, so we talk about the protocells, then we talk about the prokaryotes, if you remember. And we discussed this thing that from prokaryotes, eukaryotes, they emerge, they form, isn't it? Isn't it? Now, when you talk about the prokaryotes, I always explain this thing that P for primitive, right? P for primitive. And what is the meaning of karyon? Karyon means nucleus. So when we talk about this prokaryote, what will you see? You will see that there will be the nucleoid there. You will not see a well-defined nucleus there. I believe you all know that, right? So there is no well-defined nucleus in the prokaryotes. Rather, the genetic material is present here in the cell. The genetic material is present here in the cytoplasm directly, not having a well-defined nucleus. We call it as nucleoid, no histone protein. In the case of eukaryotes, U means true. A true nucleus is there, right? A true nucleus is there, and here you have the DNA. I hope you all are well aware of it. Yes or no? Now, why are we talking about it? I'll tell you. Here also you have the cytoplasm. Here also you have the cytoplasm, but you have other organelles also. Now, when it comes to the respiration, even the prokaryotic cell, they need energy. Even the eukaryotic cell, they need energy. So what you need to focus here? See, when you talk about this respiration, you are going to discuss the very first step here. And that very first step that we have to discuss here is glycolysis. I'll tell you why I related it with prokaryote and eukaryote. It is glycolysis. This is the very first step that we have to discuss here. What's that? It is the glycolysis. So glyco means you are talking about the glucose here. Lysis means you are talking about the breakdown here. You are ultimately talking about the breakdown of glucose here, and that too in 10 steps. Okay. But why are we talking about it? We are talking about it because in the case of glycolysis, it occurs in the cytoplasm. That is the site of this glycolysis, and it's oxygen independent. Oxygen independent. It doesn't need oxygen. Are you getting my point? So this is the first step of the respiration. So even if you talk about the prokaryotic cells, right? Even if you talk about the prokaryotic cell, because initially we know that anaerobic organism came to this earth, right? It's not just about the prokaryote and eukaryote. Initially anaerobic organisms were there, right? Because initially on earth the conditions were reducing type, right? So what happened was that anaerobic organisms were there initially, and from that anaerobic organisms aerobic organisms evolved. So in prokaryotes also, first anaerobic, aerobic prokaryotes, and then finally eukaryotes developed. So here you people can see, even if you are talking about the aerobic organism, even if you are talking about the anaerobic organism, you will see that very first step of respiration is glycolysis, which is common to both. Are you getting my point? Which is common to both aerobic and anaerobic respiration. It is common to both aerobic and anaerobic respiration. What? Your glycolysis. Is that clear? Is that clear? So yes, I started with the evolution part. I told you firstly prokaryotes came. Here also we talk about the anaerobic prokaryotes, then we talk about the aerobic prokaryotes, then we talk about the eukaryotes also, right? Then we go to what? Then we talk about the eukaryotes also, where also we will see that some organisms, they survive under the anaerobic conditions. Are you getting my point? Done. So here, what am I trying to tell you? I am saying that in the case of respiration, right, it is the glycolysis that is the first step that is taking place in cytoplasm. So look at this. If you look at the prokaryotic cell, do they have cytoplasm? Of course. In the eukaryotic cell, do they have cytoplasm? Of course. So site is cytoplasm and it is in oxygen independent manner. So somewhere you can see it on the scale of evolution as well. Right? So even in the prokaryotic organism, be it aerobic or anaerobic, first step is glycolysis. Then after that, because we know that conditions changed, accordingly the ways changed. So very first step is glycolysis, which is common to both aerobic and anaerobic respiration. So keep it in your mind. This is the first step of respiration, right? Be it aerobic organism or it is anaerobic organism. This is the very first step here. Am I clear? So if oxygen present, right? Then if oxygen present, then what will happen? It will go for oxidative decarboxylation. These are the steps that we have to study. And then finally, right? And if oxygen absent, then fermentation will occur. What will occur, guys? Fermentation will occur. And there are two types of fermentation we will have to discuss here: alcoholic or lactic acid or lactate fermentation. So that is our entire chapter. Got that? Right? So which one is the first step? This one. Site, I'll repeat it multiple times. Cytoplasm, oxygen independent, 10-step process where each step needs an enzyme. Okay? So that's what you have to study. Okay? Done. So be it aerobic organism, anaerobic organism, right? This is first here. Okay? Done? Okay, so let's get started. Let's talk about the glycolysis. But before that, let's complete this particular paragraph of NCERT as well. So I am marking NCERT right along with my lecture. So I request you all to please pay attention here, right? And please mark your NCERT. You don't need any separate session for the NCERT highlights. I'm doing it here only. So here you can see if this energy is to be useful to the cell, you know that, right? It should be able to utilize it to synthesize other molecules, right? Then the strategy that plant cell uses is to catabolize the glucose. They break down the glucose in such a way that not all liberated energy goes out as heat. Okay, so key is to oxidize glucose not in one step but in several small steps, so that beta, same story, right? So that the energy released can be coupled to the ATP synthesis. So we don't want to waste that energy as heat, so we want to trap it in the form of ATP. That's why we are doing it here. Am I clear, bache? Am I clear? So during this process, we know that oxygen is utilized, CO2, water and energy are released as byproducts. So each and every line can give you a potential and can give you MCQ. So combustion reaction, it requires oxygen, but some cells live where oxygen may or may not be available. Then what do you think? What's going to happen? So you know that it is saying that there are sufficient reasons to believe that the first cells on this planet lived in an atmosphere that lacked oxygen. Yes, early Earth, no atmosphere, then reducing condition, then finally oxidizing condition came to the early Earth as well. So even among present day living organisms, we know of several that are adapted to anaerobic conditions. Are you getting my point? So some of they are facultative anaerobes, while others are the requirement for the anaerobic condition is obligate. So in many cases, all living organisms, they retain this enzymatic machinery. They partially oxidize glucose without the help of oxygen. This is glycolysis. Now I will simplify this. Beta, there they have used the word obligate and facultative. Do you know the meaning here? Yes, please. Like here, they have used the word facultative anaerobes. I discuss each and every word here, right? Each and every word here, the mention here in the biological classification. Sometimes students say that you are teaching extra. No, I'm simplifying your NCERT. We know which terms are present where. Is that clear? Right? So let's focus here. So what is the meaning of facultative anaerobes? So before going forward, I'll tell you what are aerobic organisms. You all know that aerobic organisms are the one, they use oxygen, they need oxygen for their survival. We know it very well. Now these aerobic organisms, they are of two categories. One is obligate aerobes. What is the meaning of obligate aerobes? This is how you can remember. This is my way to remember this. I focus on this word, obligate. I remember one thing out of it, obsession. That I'm obsessed for something. I need that thing only. I don't want anything else. I need that particular thing only. That is what the obsession is. So from obligate, I remember the obsession. So obligate aerobes matlab, they need oxygen. They need oxygen only. Can't survive without it. They can't survive without it. They need it. Are you getting my point? They just need it. They cannot survive without it. That's what you have to keep in your mind. So there is another category of the aerobic organisms, that is facultative, but here you have to put the word anaerobes. Yes, you heard me right. I am saying when you are talking about the, right, I am saying that if you are done. So if I am saying obligate aerobes, I am saying they need oxygen. If there is no oxygen, they will die. They will not be able to survive. Are you getting my point? Are you getting my point? Then facultative anaerobes, you might be thinking that, ma'am, you are mentioning it incorrectly. No, facultative fluctuate. Like facultative anaerobes means they are aerobic organisms. What are they? They are aerobic organisms that need, that can even survive, not for a longer period, without oxygen for some time. So that is what I am trying to tell you. So they are not the true anaerobe, they are the facultative anaerobe. So ultimately they are the aerobic organisms only, right, which can survive without oxygen, right, for some time. They are the facultative anaerobe. Same way you can discuss what is obligate anaerobe or facultative anaerobe. I hope this part is clear. So what that portion of NCERT is telling us, that portion of NCERT is telling us that in a cell, like not all the organisms, they use the oxygen. Some are living in the condition where oxygen is not available. So if we are saying oxygen is required for the oxidation of the carbohydrate, that to glucose, what about such kind of cells? So here we are saying that such type of organisms, they start their process with the glycolysis. And in the glycolysis, what is happening? There is the partial breakdown of glucose is there, right? Partial breakdown of glucose is there. Are you getting my point? There is a partial breakdown of glucose. Now got it? Now got it? Any doubt? So let's start my favorite topic, glycolysis. Okay, yeah, genuinely it's my favorite topic, right? It's my favorite topic. Okay, okay, any doubts? Sure. Sure. So once again, glycolysis, breakdown of glucose. And this is not the complete breakdown of glucose. You can say partial breakdown of glucose. So what is glucose? Do you remember the formula? It is C6H12O6. That's what glucose is. How many carbons are there? 6. 1, 2, 3, 4, 5, 6. Now you are breaking that glucose in 10 steps in cytoplasm under anaerobic conditions, like, or I would say under oxygen independent conditions. So what you people will see, this is what you guys are going to see. You're partially breaking it. So that's what you are going to get here. This is what we have to understand at the end, that this is the glucose, a 6 carbon containing compound. So we will break it into 2 3 carbon containing compound. And what is it, beta? What is it? That is, yes everyone, that is, quick, pyruvate. So pyruvic acid, ionic form is pyruvate. So these are 2 pyruvate, 2 3 carbon containing compounds. Okay, 2 3 carbon containing compounds. Now, to start this process in detail, first of all, each and every step needs enzyme, and the cofactor used here is magnesium ion. So firstly, I will not write, जैसे देखो glucose, will tell you glucose 6-phosphate. I'm writing few names. Fructose 6-phosphate. Beta, then I'll explain everything. Let me write few words. Fructose 1,6-bisphosphate. Glyceraldehyde 3-phosphate. Dihydroxy acetone phosphate. Then for glyceraldehyde 3-phosphate, we'll use the short form G3P or PGAL. We'll explain it. This is DHAP. Bis-phosphoglycerate, 3-phosphoglycerate, 2-phosphoglycerate, phosphoenol pyruvate. I'll explain everything, just allow me some time. And then pyruvate. So these are the names in front of you. Okay. Hanji, understood? These are the names which you have to remember first. Quick. These are the names which you have to remember first. I'll start directly with the glycolysis. After that, there are just a floating respiration, protoplasmic, aerobic, anaerobic may difference. Then I will cater it, right? Because that are very general things. So firstly, we'll talk about the glycolysis. So tell me, this part is clear? Like, I'm just making it easy for you. Firstly, I want you people to understand that what exactly is happening. Okay? Okay? So you have to remember it. You have to remember it. Give it a try. And as I said, after that, I have to discuss few definitions. Just floating respiration, already told you about the protein, protoplasmic about the proteins. So we'll discuss it. Yes, please. Quick. Hanji, quick. Glucose, glucose 6-phosphate, fructose 6-phosphate, fructose 1,6-bisphosphate, glyceraldehyde 3-phosphate, dihydroxyacetone 3-phosphate, then you have 1,3-bisphosphoglycerate, 3-phosphoglycerate, 2-phosphoglycerate, phosphoenol pyruvate. First of all, we have to remember it. If it is done, very good. If not, please take time. Please take at least 2 minutes and try to remember it, right? At least give it a try. Yes, everybody. At least give it a try, please. Glucose, glucose 6-phosphate, fructose 6-phosphate, fructose 1,6-bisphosphate, glyceraldehyde 3-phosphate, dihydroxyacetone phosphate, then 1,3-bisphosphoglycerate, 3-phosphoglycerate, 2-phosphoglycerate, phosphoenol pyruvate and pyruvate. Let's repeat again. Glucose, and we'll make a short on that too. So what are we talking about? Glycolysis. How many steps are involved here in this particular process? 10 steps. Is it the complete breakdown of glucose? No, it's the partial breakdown of glucose. What is happening here? Basically, breakdown of glucose into 2-pyruvate, that too in cytoplasm in oxygen independent condition. So it's a common step for both aerobic as well as for the anaerobic respiration. So here there will be the breakdown of glucose. There will be, firstly, what will happen? Glucose will form glucose 6-phosphate, then fructose 6-phosphate, fructose 1,6-bisphosphate. Then after that, what will be there? There will be the breaking down of that, right? Then the cleavage basically. So glyceraldehyde 3-phosphate and dihydroxyacetone 3-phosphate will be there. Then dihydroxyacetone 3-phosphate will be converted to G3P. Then G3P, it will form 1,3-bisphosphoglycerate, 3-phosphoglycerate, 2-phosphoglycerate, phosphoenol pyruvate, and finally pyruvate. Right? I want you to remember this, and then I'll tell you the logic behind, the steps behind, everything. But firstly, so who's so I'll give you, it's 8:26, 8:28. Try to remember it. I know you can do that. You're Gen Z people, quite smart, quite fast. Do it quickly. Yes, do it quickly. Chalo, remember that, quick. It is in front of you, do it. It is in front of you, do it. Then I'll simplify it. Then I'll help you out and I'll tell you why I am writing this here. Why are we writing this here? And not just this, there are many other things that I need to add here. But firstly, remember the names. Done? Pakka? Sure? Sure? Take your time, do it. We have many things to add here. We have many things to discuss here. But firstly, tell me this part is clear. Yes. Okay, chalo. So let's go for it. So as I said, this is the glycolysis, partial breakdown of glucose. You are breaking a 6 carbon containing glucose into the 3 carbon containing compound. So basically here they all are one by one. We'll go so that we can just simplify it. That's why this is a 3 carbon, 3 carbon. Then they all are 3 carbon containing compounds. Got that? This is what we have. Now in the glycolysis, right students, it occurs in 2 phases. In the glycolysis, we will talk about number 1. It is going to be the preparatory phase. What will be there? Preparatory phase. Number 2 will be the payoff phase. Preparatory and payoff. Preparing. Like I'll give you the example. Like let's suppose you want to start a business, right? You want to start a business. First you need what? You need the idea and obviously then the investment. In the preparatory phase, what will happen? ATP will be used. In the payoff phase, ATP formed. It's like first you are investing money, then you are making money. It's like that. So when it comes to the preparatory phase, first 5 steps, how many steps guys? First 5 steps they are making the preparatory phase. Next 5 steps they are making the payoff phase. They are doing what? They are making the payoff phase. This is happening. So now we have the glucose in the cytoplasm. So what is happening here? The glucose is converted to the glucose 6 phosphate. So here phosphate is added at the 6 carbon of the glucose. So we use the enzyme hexokinase here. Magnesium ion is used as a cofactor. But from where are you going to give phosphate here? You will use the ATP, adenosine triphosphate, that will make adenosine diphosphate. So which enzyme are we using? Hexokinase. Wherever you will see the kinase, you will find out that this enzyme has something to do with the phosphate. Right, that's how you can remember. This enzyme has something to do with the phosphate. So hexokinase, 6 carbon hexose, right? And no doubt, at some places you will read about the glucokinase here. I will clarify that detail also, students, but not right now. Right now, one by one we are going. So you are doing what? You have the glucose, you are converting that glucose into the glucose 6 phosphate. Can you see there is only one arrow? This diagram in NCERT is given incorrectly. So pay attention. So glucose, what that glucose is making, I told you glucose is making glucose 6 phosphate, and this is an irreversible step. I'll tell you why. Which enzyme are you using? Hexokinase. Now, from glucose 6 phosphate, you are making the fructose 6 phosphate. We know that. What are they? They are the isomers. So here the enzyme is glucose phosphate isomerase, right? You can also call it as phosphoglucoisomerase or glucose phosphate isomerase. Then again, fructose 6 phosphate, and here you have fructose 1,6 bisphosphate. Now what does it mean, students? See, if there is a compound, like let's suppose 1, 2, 3, 4, 5, 6, there is a 6 carbon containing compound. If there are 2 phosphate attached to the same carbon, just here also, then you will use diphosphate word. What is the term that you are going to use here? You will call it as diphosphate. But now here what is happening? So it is 1, 2, 3, 4, 5 and 6. So here you people will see one phosphate is at, this is 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. So one phosphate is at first carbon, another phosphate is at 6th carbon, right? So 2 phosphates are there in the compound but are attached to different carbon. You are using the word bis here. So just say here we are saying, right, here we are saying that what is happening? We are saying glucose will form glucose 6 phosphate. ATP is used here. So it will give its 1 phosphate here and we will get the ADP. Enzyme use this. Magnesium ion is used for all the steps here. All the steps of glycolysis. Then fructose 6 phosphate. It means the phosphate group is attached to the 6 carbon. Now here, how many phosphates are there? 2. Fructose 1,6 bisphosphate means to 1 first carbon and to the 6th carbon. Now here again you are adding phosphate. So from where are you getting it? You are getting it from ATP. Again you will have the ADP. I'm not discussing why, this is regulatory, this is the locking step. This is the regulatory step. I am not concerned about it right now. I want you to understand this portion. then this portion, then I will tell you all other details here. So any doubts? Any doubts? Hanji, any doubts? Yes? Sure? Up to this part all clear? Up to this part all clear? Sure? So again, here also you are using the ATP. Now, here the enzyme is phosphofructokinase, a very, very important enzyme. Right? A very, very important enzyme. Clear? Are you sure? Are you sure? Okay. Then fructose 1,6-bisphosphate. This is the 6 carbon containing compound. You are breaking it. So here also you are having 3 carbon containing compound. Here also you are having 3 carbon containing compound. So here what is happening? The cleavage. See, they say here it is phosphorylation, isomerization, phosphorylation. Now here what is happening? Cleavage. So which enzyme is used here when you have to break it? This is the aldolase, right? Aldolase will be used here. So it is breaking it into G3P, which is glyceraldehyde. What is it? Glyceraldehyde. Remember carbohydrates. When you talk about the carbohydrates, you talk about the monosaccharide, you firstly say triose. Just if you talk about the monosaccharides, that is the single sugars. So you have triose, you have tetrose, you have pentose, you have hexose, haptose. That's what we discussed there. Basically, in the monosaccharide, you see the carbon number is from 3 to 7. 3 to 7. So, in the monosaccharides, we know it. Aldehyde group is there, aldose. Ketone group is there, then it is ketose. So, if you remember, we discussed the glyceraldehyde. So, it is the monomer, a monosaccharide having 3 carbon. Right? It is the aldose. Its functional group is aldehyde here. And now, if it is just a dihydroxyacetone, this is again a 3 carbon containing compound. This is again a 3 carbon containing compound. This is also a 3 carbon containing compound. So glyceraldehyde mein aldehyde is the functional group. Here ketone is the functional group. You know that aldehyde, this functional group is the terminal. This is subterminal one, not discussing their properties, but just telling you this. So they both are 3 carbon containing. So glyceraldehyde 3-phosphate dihydroxy. So it is aldehyde. What's the functional group? It is aldehyde, right? CHO, that is the functional group, right? Aldehyde functional group is there. So I have seen students making this mistake. So please pay attention. If it is written as glyceraldehyde 3-phosphate, if it is written as glyceraldehyde 3-phosphate, that is G3P, you can also write it as PGAL. Now what's that? That is phosphoglyceraldehyde. What is that, beta? That is phosphoglyceraldehyde. What's that? That is phosphoglyceraldehyde. Is that clear? Is that clear? So just keep it in your mind. Just keep it in your mind, number 1. Now, if it is written as 3-PGA, that is different. Do not confuse PGAL and 3-PGA. It means 3-phosphoglyceric acid or glycerate, whatever you want to write it. So if the short form is written as 3-PGA, you are talking about the carboxylic acid, acidic group here. What are you talking? You are talking about the acidic group here. What are you discussing here? You are talking about the aldehyde. Am I clear? You're talking about the aldehyde here. You're talking about the acidic group here. So PGA is different. PGAL is different. Am I clear here? Right? Am I clear here? Quick. Quick. Done, beta? Sure? So, Glyceraldehyde, they both are also isomers. So here, which enzyme do you have? You have triose phosphate isomerase. Triose phosphate isomerase. And as I said, magnesium ion will be used in all the steps. Now, so far, if there is any doubt, then tell me, quick. So far, if there is any doubt, then do let me know, quick. Guys, quick, I'm waiting. Done, beta? Noted? Done? Beta, you are breaking it, aldolase. It is the cleavage. So you are breaking it into 3 carbon, 3 carbon, 2, 3 carbon. It's like you have 6 chocolate, you are distributing 3 to 1. First kid, 3 to another kid. It is like that. So now if this part is clear, then first step, second step, third step, this cleavage is the fourth step, this isomerization is the fifth step. So it is the preparatory phase that I have discussed here. Don't worry, I have many good flowcharts. Right now, I want you to understand this portion. This is the preparatory phase. Am I clear here? Yes? Any doubts? Any doubts? Quick. Hanji, sure. So now from this part, can I conclude here that when I'm talking about the preparatory phase, guys, I'm going very slow, like you are reading it for the first time. We have step 1 to 5, number 1, right? I'm writing the conclusion here. I am saying step number 1 and 3 are irreversible and ATP is used here. Am I clear? ATP is used here. Am I clear? Am I clear? Step 1 to 5, 1 and 3 are irreversible and ATP is used here. ATP is used here. Am I clear? So this is the story of first 5 steps. This is the story of first 5 steps. Now let's go to the 6th step. Let's discuss about the 6th step. Now what is happening here? You have glyceraldehyde 3-phosphate. You have dihydroxyacetone 3-phosphate. Actually, what is happening? Once this G3P and DHAP form, right? You understand chemistry better than me, I guess. Right? This DHAP will also be converted into G3P. I'm repeating myself again. Glyceraldehyde 3-phosphate, dihydroxyacetone 3-phosphate. Obviously, ketone, it is having the functional group ketone. This one is having the aldehyde. what is happening? This reaction dynamics are in such a way, this DHAP will be converted to G3P. So now instead of having one G3P, how many G3Ps do we have? We have two G3Ps. We have two G3Ps. Tell me, we have two G3Ps. Is that clear? Is that clear? Quick. Tell me. Firstly, tell me this point clear, everybody. Quick. So DHAP will get converted to G3P because it is the G3P only that can go for the payoff phase. Why is it so? Why it is the G3P only? Because that enzymes are specific for this only. Your dihydroxyacetone 3-phosphate cannot enter in the payoff phase. These enzymes are specific for the G3P only. So if we will not convert this DHAP into G3P, then obviously 3 carbons of this glucose, this part will get wasted. We don't want that. So DHAP will get converted to G3P. Then how many G3Ps do we have? 2 G3Ps. How many G3Ps do we have? 2 G3Ps are there. Now, again, this is the 6th step. This is the payoff phase. It is the phase where ATP will form. Now you are not using it. Now you are making 1,3-bis-phosphoglycerate from it. Yes or no? Now you are making 1,3-bis-phosphoglycerate from it. 1,3-bis-phosphoglycerate from it. Am I clear? So either you can write it in this way, multiply 2, or you can do this also. Your choice. Okay? I'm repeating this part again. Either you can write 2 multiply 1 thing like 2 these compounds, or you can put it in bracket. It's one and the same thing. Right? Because we know that now we don't have 1 G3P, we have 2. So whatever you are going to take further, you will take it as 2, 2, 2, 2, 2. Just say, see, now 2 into G3P. This is what we have. But here you are not using the ATP. Then how will you arrange one extra phosphate? So this time it is the inorganic phosphate which is used. This phosphate is not taken from the ATP, but the inorganic phosphate is used. You can write it in this way also. But how many? Because 2 times the step is going on. So you can say that 2 pi. Right? 2 pi. Now here, your NADH. Okay, so this NAD, nicotine-amide adenine dinucleotide, it is getting reduced. NAD, it will form this. So 2 NAD will give you 2 NADH2. Are you getting my point? So here oxidation. So here what is taking place? Phosphorylation hai, dehydrogenation hai. Haan. So NADH2 will form here. NADH2. We'll explain it. Right? We'll explain it. Then this is the 7th step. 7th step. This is beta 3-phosphoglycerate. A very, very, very important step. And here what is happening? Because we have to see now that this step is taking, because 2 G3P are there. That's why I'm multiplying here with 2. So here beta 2 ADP will give us 2 ATP. This is again the irreversible step. Again the irreversible step. So we have G3P, it is giving us 1,3-bis-phosphoglycerate. You can call this, and this is the 1,3-bis-phosphoglycerate. So this is the phosphoglycerate dehydrogenase enzyme as well. Right, so what is it? It is the phosphoglycerate dehydrogenase enzyme as well. Which enzyme is there? Phosphoglycerate, or you can simply say glyceraldehyde 3-phosphate dehydrogenase. That's the enzyme which we are using here. Just a minute. glyceraldehyde 3-phosphate dehydrogenase. Understood? Glyceraldehyde 3-phosphate dehydrogenase. Now 1,3-bis phosphoglycerate will give you 3-phosphoglycerate. So now here you have one less, right? Now here what do you have? You have one less phosphate, isn't it? So where that phosphate will go? That phosphate, right, that phosphate will go to what? That phosphate, 1,3-bis phosphoglycerate. Now it is 3. So this phosphate will be passed to this ADP and ATP will form. So which enzyme is used here? The enzyme used here is phosphoglycerate kinase. Phosphoglycerate kinase, guys, and this step is substrate level phosphorylation, a very, very important topic here. So what is the substrate level phosphorylation? Right, what is this substrate level phosphorylation? So substrate level phosphorylation, first of all understand the phosphorylation. So phosphorylation means addition of phosphate, right? Phosphorylation means addition of phosphate. And when it is the substrate level phosphorylation, then what is happening, beta? Then what is happening? So basically here, what is happening, guys? What is happening? Just see, here oxidation and phosphorylation is going on. Here you will see what is happening here. Substrate level phosphorylation is occurring. Phosphorylation means addition of phosphate to ADP, right? When inorganic phosphate is added to ADP and you get the ATP, this is what we call as phosphorylation. Are you getting my point? This is what we call as phosphorylation, that is the addition of phosphate. Now substrate level. So here what is happening? This substrate itself is giving this phosphate to the ADP, you are getting the ATP here. So it is the substrate level phosphorylation. Are you getting my point? Irreversible step. Now this 3-phosphoglycerate will give me 2-phosphoglycerate, and this is simply the rearrangement, nothing else. Just say for an example, if 1, 2, 3, third carbon is having the phosphate. Now here you are doing 1, 2, 3, second carbon is getting the phosphate. So this is the rearrangement, simply the rearrangement. So the enzyme used here will be mutase. What is the enzyme which will be used here? That will be mutase. Am I clear? Am I clear? Now this one, this 2-phosphoglycerate will give us phosphoenol pyruvate. Done, beta? Phosphoenol pyruvate. And then here you will see, then finally there is the pyruvate. So here the enzyme used is enolase, here the enzyme used is pyruvate kinase. But what we have to see here, so look at this, 3-phosphoglycerate will give us 2, then 2-phosphoenol pyruvate, and finally phosphoenol pyruvate to pyruvate. Where is that phosphate? Again here also 2 ADP will be converted to 2 ATP. Can you see that? 2 ADP will be converted to 2 ATP. This is again the substrate level phosphorylation. Am I clear? So in the payoff phase, it is in front of you. You can see that very clearly. Basically step 1 and 10, irreversible. There are many things which I have to still clear here. They are irreversible, number 1. And it is the 7 and 10 where what is happening? Where substrate level phosphorylation is occurring. Where substrate level phosphorylation is occurring. Where what is occurring, beta? Substrate level phosphorylation. So in totality, how many ATPs form? How many ATPs formed here? You can see that they are 4 ATPs. Why 4? Because we know that this G3P, DHAP is converted into G3P, that is why. Are you getting my point? That is why. So that is what you have. But this is not the total, of course. Now, it's like for starting a business, let's suppose, you took 10 lakh from somebody. For starting a business, let's suppose you took 10 lakh from somebody, right? You took 10 lakh from somebody. Yes or no? Yes or no? Then you, now after that you made a profit of 20 lakhs, but you have to return that 10 lakh now. So your net gain will be 10 lakh only. Same is the case here. You made 4 ATP, but how many ATP you used? 2. So 4 minus 2 will be 2 only. So this is your gain. Plus you also made 2 NADH2 as well. So it is nicotinamide adenine dinucleotide phosphate, right? Right? So I'm not done here, but firstly tell me, is that clear? Is that clear? Because now we have to discuss the dynamics of enzymes as well. Oh, I didn't add the diagram. Chalo. So beta, this is the diagram, the flowchart that I made na, that is what you have to accept, the one that I made. Okay, the one that I made, that you have to accept in the NCERT, that arrows are not proper. Okay. Okay, so ye dekho, glucose 6-phosphate, irreversible, ATP is used. Fructose 6-phosphate, you can see phosphohexose या phosphoglucoisomerase. Fructose 6 to fructose 1,6 bisphosphate. Again you have used the ATP. Then fructose 1,6 bisphosphate to glyceraldehyde 3-phosphate dihydroxyacetone. So aldolase is used, cleavage is taking place. Then we know that this is also the reversible step. Then this G3P, DHAP will be converted to G3P. Enzyme used here for the 5th step isomerization is triose phosphate isomerase, told you already. Then why G3? Because rest all the enzymes are specific for the G3P. So 2 times it is. So yes, that's how you write the inorganic phosphate. So to these NAD, this is oxidized. So it is reduced here to NADH2. So nicotinamide adenine dinucleotide phosphate. So oxidation and phosphorylation is occurring. 7th step again, irreversible. So 2 ATP are formed. This is the substrate level phosphorylation from 1,3-bisphosphoglycerate. You have 3 phosphoglycerate. So this phosphate will go here. So 3 phosphates to 2 phosphate. So again, rearrangement. So you can see the enzyme is mutase. So phosphoglycerate kinase, phosphoglycerate mutase. So wherever you will find the enzyme mutase, it is for the rearrangement. Then 2 phosphoglycerate to phosphoenol pyruvate. Water is released here, right beta? So enolase is the enzyme, finally pyruvate kinase, then this is again the substrate level phosphorylation. Okay? Structures are also given there. So if somebody will ask me question, I'll tell you what you have to prepare from this. How many ATPs and where? Like you have to remember the reactions here are used. Number 1. Where ATP formation occur with reactions, you have to remember. How many, same question it is, steps of substrate level phosphorylation. What is substrate level phosphorylation? Which one are the regulatory enzymes? Net gain here, fate of pyruvate. These are the kind of things that you can ask from yourself. Are you getting my point? Done? That's what you have to remember. Now, look at this. This first step, very first step, do you know that it is known as the locking step? Do you know it is known as the locking step? Quick. Done? Hanji. We'll discuss it further. Tell me. Guys, quick. Hanji. Why it is so? देखो बेटा, जैसे let's suppose if somebody is in hospital, right? you know that in our case also, we also have to take the energy. This is actually a question. It's not something that I'm making. If somebody is hospitalized, then why? Matlab, why? Like, you know, na, in that solution, we have that saline or this glucose and all. I hope you are aware of these general things. So why can't we directly give to that patient the glucose 6-phosphate? Why are we giving that patient glucose? Patient is already weak. Patient is already weak. So why are we giving that patient glucose first? Why can't we directly give glucose 6-phosphate? Because if you are going to give glucose to that patient has to convert it to the glucose 6-phosphate first, right? Patient has to convert it to what? Patient has to convert it to the glucose 6-phosphate first. So why can't we directly give that person glucose? It's simple, beta, because this glucose 6-phosphate, it is impermeable to cell membrane. Are you getting my point? It is a locking step because when inside the cell cytoplasm, when you convert this glucose to G6P, then it will be trapped here only. We can carry it forward for the further steps. Are you getting my point? We can do that. Are you getting my point? Yes? Yes, thank you so much, beta. Thank you. Right? No, it is not the simplest form. That is the reason. That's why you are doing it. Is that clear? That's why you are doing it. Is that clear? Sure? So this is the locking step. Now, this enzyme is very, very, very important here, fructokinase. This is the main regulatory enzyme. Even hexokinase, pyruvate kinase is also the regulatory enzyme. But if somebody will ask you main regulatory enzyme, then it is the phosphofructokinase. So now I will tell you why. But firstly, tell me these points are clear. Sure? These points are clear. Now I'll show you something very amazing. My handwritten notes. Trust me, my handwriting is quite good. Chalo. So I'll come up with the glycolysis, because you'll be amazed to know, right? You will be amazed to know that there are so many beautiful things about this topic that we don't even discuss, and I'll show you. Somewhere it is in NCERT, it's just that we do not focus there, right? We do not focus there. Okay, so glycolysis, who discovered it? Ambika Ma'am, Parnas pathway. So it is also known as EMP pathway. It is also known as EMP pathway. So glycolysis, coming from the Greek word glycose, sugar, lysis, splitting. So in 1930, Ambika Ma'am Parnas has given this sugar splitting pathway. Site told you cytoplasm oxygen independent process. Guys, trust me, nobody is going to explain a topic 3 times in a lecture so that you can clearly retain it, right? So just value this thing and revise it properly, okay? Okay? Done. So in all living organism, whether it is of aerobic or anaerobic type, the first is the cellular respiration is glycolysis. 10-step process where partial breakdown of glucose is there, not the complete breakdown here, right? Complete breakdown of glucose will only be there in the oxygen presence. So here you can see it's a common phase. So in plants, glucose is derived from the sucrose. If you know it very well, otherwise, in the plants, remember, it is the phloem that will help in the translocation of the organic food. So phloem is having the sucrose. Are you getting my point? Phloem is having what? Phloem is having the sucrose. Sucrose, we know that it's a disaccharide. It is an invert sugar. It's a disaccharide. It is an invert sugar. It's a disaccharide. It is an invert sugar. Are you getting my point? So made up of glucose and fructose. So plants, they will take their glucose from this sucrose itself, right, students? So we know that sucrose, glucose, fructose, that if glucose, then it will enter in this glycolytic pathway. Now here, guys, trust me, it's going to be amazing for you, right? So see this, as I said, preparatory and payoff phase is there. 5 steps are there. So in preparatory phase, ATP will be used. In payoff phase, ATP will be formed, right? So why I just said that it is the phosphofructokinase, yeah, you write it as PFK1. That is also the way to write it. It is basically the main regulatory enzyme. Just now I told you guys, yes or no? It is also known as pacemaker of glycolysis. What do you call it? You also call it as pacemaker of glycolysis. Are you getting my point? Pacemaker of glycolysis. Are you getting my point? Yes or no? Yes or no? Why? Because it is inhibited by high concentration of ATP and citrate. If in the cytoplasm, if in that cell, if there is high concentration of ATP, if there is high concentration of citrate, especially let's talk about the ATP. High ATP means already the energy is there available. So what is the need to go for the respiration then? So high concentration of ATP will inhibit this, and ADP and inorganic phosphate will stimulate it. Are you getting my point? Will stimulate it. Is that clear? Is that clear? So this phosphofructokinase 1 is the allosteric enzyme, guys. It is having this enzyme, we know that they are proteinaceous, they are having a dimer. So one half of this dimer beta, it is having the ATP binding site. Another half can bind to the substrate, right? Allosteric enzyme it is now, right? So, and a separate allosteric binding site, right? One is for what? One is for the ATP. Another is for the substrate, and there is a separate site where ADP or AMP can join. So if there is high ATP, it will stop the glycolysis. If there is high ADP and inorganic phosphate, then it will stimulate it. If there is high ADP or AMP, it will be stimulated. Main regulatory enzyme. So these two are also the regulatory enzyme, but which one is the main? Your phosphofructokinase 1, right? ATP and citrate. Yes, you have to remember, important. So hexokinase, it is inhibited by excess of glucose 6-phosphate. जैसे ये देखो, glucose is making glucose 6-phosphate. So it can itself prevent the activity of this hexokinase. And what about this? Once this phosphofructose kinase, once this fructose 1,6-base phosphate form, then to it's a go ahead signal ki go for the step. Then this pyruvate kinase, right? ATP. So here for pyruvate kinase also ATP and acetyl coenzyme are the inhibitor, and fructose 1,6-base phosphate will act like an activator, because once it is formed, then it is a signal, right, that now to we have to carry forward the step, right? Now another question that I asked you, hexokinase or glucokinase. I told you that for the first step you can also use glucokinase, you can also say hexokinase, right? Generally we will say hexokinase. Why is it so? Very interesting explanation related to your Michaelis-Menten part, and I believe nobody will discuss that in that much detail, beta. Nobody will discuss that. Okay, right? Sure. Acha, so we have the spammers here. Okay. Acha, this session is having live 100K, beta. It doesn't matter to us. Right, we have seen 100K plus live or even 10K plus live. We know that, right? And we know the quality of the content there. So we have no need to compare our channel and our teaching's quality with any other platform or with any other teacher. So I'm here to teach. Those who are here to learn, most welcome. Those who are not, tada, bye. Chalo. So hexokinase. Next is what? Hexokinase. Beta, this enzyme you will mainly get in brain cell or muscle cell. Glucokinase will be mainly in the liver and pancreas. I'll tell you the reason. This particular enzyme, it is found nearly in all cells. This is having high affinity for glucose. Now, madam, what is the meaning for high affinity for glucose? If an enzyme is having high affinity for its substrate, then if its substrate is available in less quantity, less amount, still it can bind to that substrate, even if it is available in less amount. And if an enzyme is having low affinity for the substrate, then you need that substrate in more quantity so that it can bind to the enzyme. So here you can see this one is found nearly in all cells. It is having high affinity for glucose. So high affinity mane Km value, right? Km is what? Km is that substrate concentration at which the velocity will be half maximal. So high affinity matlab low Km means low Vmax of that reaction. And here in the liver and pancreas, this enzyme is having low affinity for the glucose, that is high Km value, because if there is high affinity for the glucose, even if glucose is in less quantity, the reaction will start, the reaction will proceed. So you need less substrate concentration at which velocity will reach half maximal. So it can act, this glucokinase can act on other 6 carbon sugars as well. It is like, okay, if glucose is there, I'm very happy. If not, chalo, if there is any other hexose, I will act on it. So this enzyme is inhibited by G6P. Now in liver and pancreas, you know that liver is a place where the glycogen will also form. Glucose, when there is glucose, the excess of glucose is converted to what? To glycogen. Now if imagine, if in liver, if this enzyme will have high affinity for the glucose, so this enzyme is going to act on the glucose, will start breaking it down. But in the liver, we don't have this requirement. We want the glucose to get stored, and if there is excess of glucose, then after the storage in the form of glycogen, then we are okay, theek hai, there should be the breakdown. That is why. Are you getting it? So that is why here such type of conditions are there, right? So that is why, and this glucokinase is not inhibited by G6P. So that's the detail of the topic which I have given you in detail. One more detail is there. So chaliye, done. So step 1, 3, 7, 10 irreversible, 7, 10 substrate level phosphorylation. So it's a metabolic process that directly produces ATP by transferring a phosphate group from high energy organic molecule to ADP. Understood? Understood. So you should also know where that NADH2 is also formed. So here also I have added this. Now some students, not some students, a particular student asked me question regarding this step. Wait, I will show you. There is a question, I got a question from one of my students for this particular part. Let me show you. For this particular step now, one of my student has asked me the question. Right, that ma'am, this is not reversible, this is irreversible. I'll discuss that with you all. But firstly, tell me, up to this part all clear? Hanji, first of all, tell me, this part clear? Sure? Quick, then I'll discuss one more question. Yes, please. चलिए, so one of my students asked me a very wonderful question from me, जैसे phosphoglycerate kinase, short form is PGK. जैसे one of my student asked me that, ma'am, here you said that 7th step in the glycolysis is the irreversible. It was a very good question, I really liked it, but when you will understand the depth of it, then you will be able to get to know about it more. So beta, it's catalyze a reaction which is, I would say, near equilibrium, right? I would say it is near equilibrium, right? When you talk about this reaction, this enzyme, this catalyzer reaction, which is near equilibrium, and yeah, we do know that it is basically going to form the reversible reaction. But what exactly is the thing? In the case of glycolysis, because this is feasible, here, this enzyme, it is affected by the substrate as well as the product concentration. I am not saying that in glycolysis it is the major regulatory step. Now, here you will talk about phosphofructokinase. But here, what's the story? What's the point? As I said, it is near equilibrium, nearly equilibrium. Baki, this is affected by substrate and product concentration. In the case of glycolysis, right, thermodynamically it is possible. In the case of glycolysis, we need this thing, this 1,3-bis phosphor, because here substrate level of phosphorylation is taking place. ATP formation is there. So here 1,3-bis phosphoglycerate will form 3-phosphoglycerate. But if you will discuss the same enzyme in gluconeogenesis, what is gluconeogenesis? Gluco means glucose, neo means new, genesis means formation. So when we make the glucose from a new source, from a non-carbohydrate source, in that case also, when you talk about this enzyme, then it catalyzes what? Then it catalyzes the reversible step, right? So it depends. Is that clear? Done? So is that clear? Is that clear? That why here I'm showing you the irreversible thing? Yes. So in RBC, because RBC, it solely depends on the glycolysis for its energy requirement. Is that clear? It solely depends on that, right? It solely depends on that. So then this is a very crucial step. So remember, 1,3-B, this bis-phosphoglycerate is having the high energy beta, right? It is having what? It is having the high energy. Okay. So high energy acyl phosphate bond. So here you can see phosphate transfer to ADP is thermodynamically feasible. So I told you when and where this thing will occur. So this is all about the glycolysis, right? And after this, I'll teach you some general terms of the, right, some general terms of the respiration, and then we will start with fermentation. So any doubt? Any doubts? So let's have a 5 minutes ka break. 5 minutes means 5 minutes this time. Let's have a 5-minute break. Only 5 minutes. Okay? Because we will finish it early. It's not a lengthy step. So only 5 minutes ka break you guys will get. Hello. Chalo. Let's start the next part. Am I audible? So next part is the fermentation. And now answer these questions. Hanji, now answer these questions. Quick. Answer these questions. Quick. Do you have any answer? How many ATPs are formed? Total 4, but we have to minus 2 as well, which we have used in the preparatory phase. And how many NADH2? 2 NADH2 formed, right? So that is what you have to remember. And in the ETC, we will see what will we do with this NADH. Okay. So beta, this is what they have given. Okay, so here there is a question that can you then calculate how many ATP molecules are directly synthesized here? So directly synthesized are 2, and we will discuss this electron transport chain, and there you will see that this NADH2, right? That will also convert into the ATP in ETC. So that we will discuss later. Am I clear? Okay, so now the next part is we'll see the fate of this pyruvate. But before that, I would like to discuss this thing. वैसे you already know about that. In the last class also I told you about the external, internal and the cellular respiration, if you remember, right? So here you can see external respiration. It is the exchange of respiratory gases between organism and environment. Remember in the case of human lungs, I told you when there is an exchange in between the alveoli and the blood capillary. Then internal respiration, it is the exchange of respiratory gases between tissue and extracellular environment. Then cellular respiration, enzymatically controlled, multi-step process. Got that? These 3 definitions clear? Right? These 3 definitions clear? So here the next part is so fats are used as respiratory substrate by number of organisms because they contain the energy, more energy in comparison to carbohydrates. Already told you they are not directly used in respiration because they are first broken down into intermediates. We can never use, this is very important, we can never use fats directly into the respiration because we have to convert them into acetyl coenzyme or glyceraldehyde, etc. Then what is protoplasmic respiration? It occurs in the starved cell. When cell is under starvation, when it is like when the energy sources are not available, these other respiratory substrates are not available. So in that case, cell will start digesting protein. And I told you when cell will start doing this, then it is very risky, right? Why? Because all that we are basically proteins, right? They are making this matrix, they are making the enzymes as well. So in that case, that will also be digested. So proteins which are rarely used as a respiratory substrate, and when during seed germination of protein rich seeds and spores. So I am saying that this respiration, it depletes the protoplasm, right? It depletes the protoplasm, and though that protoplasm is having structural and functional proteins which liberates toxic ammonia. So this is the protoplasmic respiration. Understood? Baki differences in aerobic and anaerobic we'll discuss at the end, and I hope this glycolysis part is clear. So this is the reaction, beta. This is the reaction. This is the reaction of glycolysis, which should be very clear in your mind. Is that clear? Is that clear? So 1 glucose, 4 ADP, 2 inorganic phosphate, 2 oxidized NAD, 2 ATP are used. Enzymes and cytosol are used. So 2 pyruvic acid, 4 ATP, 2 NADH2 and 2 ADP are formed. Is that clear? Is that clear? Sure. So this is the flowchart, students. This is the flowchart. Please have a look here. You can see it very clearly that glucose 6-phosphate can have negative feedback on hexokinase, ATP, AMP, even along with that citrate. They negatively inhibit PFK, phosphofructokinase and pyruvate kinase. Am I clear? Sure? Are you sure? So the next topic that we have to discuss now is the fermentation. What are we going to talk about? Now we will talk about the fermentation. Ready? Ready? Okay. So here in fermentation, see, firstly, how will it start? Glucose will form 2-pyruvate. है ना? That pyruvate, if oxygen present, then it will go to mitochondria. For what? For oxidative decarboxylation and even for Krebs cycle and then etc. Now, if this is absent, I told you already, it will go for, yes, my dear students, it will go for fermentation. Where will it go? It will go for fermentation. Am I clear? So that is what we have to discuss. We will talk about the fermentation. Understood? We will talk about the fermentation. So in fermentation, say by yeast. What is yeast? Yeast is a unicellular fungus belong to class Ascomycetes. Okay. So what exactly is the fermentation, beta? It is the incomplete oxidation. It is the incomplete oxidation of glucose and it will give us very less energy in comparison to the complete oxidation of glucose. Is that clear? Is that clear? Is that clear? Sure? Beta, there is a power cut, so I will see that for how long, you know, this backup can survive. If there will be, if all of sudden, right, if class ended all of sudden, then be clear that it is because of the power issues. So I'll take the part 2 tomorrow in the afternoon, if there will be anything like that. Otherwise, there is no chance. I really want to finish it today only, right? And I know I'll be free by 11. Chaliye. So now the next part is, so under anaerobic conditions, right? So under anaerobic condition, what is happening? Right, sets of reactions will occur where pyruvic acid is converted to CO2 and ethanol. So ethanol, mane you are talking about the alcohol here. So that's the diagram. I'll start explanation with this particular diagram. This is important. You can see glucose. This is showing further steps here. We know that when glyceraldehyde 3-phosphate, when we are getting 3-phosphoglyceric acid. So it is glyceric acid, just a carboxylic acid, or carboxylate. When I'm writing this, it in the form of this 8. So we are talking about the ionic form. What are we talking? We are talking about the ionic form. So you can see NAD is oxidized. It is reduced. So here you can see then phosphoenol pyruvate. You have to remember this flowchart like this. You have to remember. It is important, guys. Then see pyruvic acid. So can you see this is one side, this is another side. Look at this diagram. Firstly, I'll explain this to you. It is the lactic acid fermentation, right? And this one is the alcoholic fermentation. If you look at the lactic acid fermentation, there's a fermentation mein, two types of fermentation is there. The products of fermentation, they can be same, they can be different. Like we can have the homo fermentation or hetero. Hetero mein you will get different products. Now, when pyruvic acid is getting converted to lactic acid, if you will notice here, one arrow only. And what is happening here? Here, beta. See, do not confuse these two things. Here your NADH, NADH2 is oxidized. In the glycolysis, we discussed that 2 NAD positive, they will be reduced, they will form NADH2. Here your NADH, it will be oxidized. So one arrow, one step process, lactic acid fermentation. The enzyme used is lactic acid dehydrogenase. Now look at this diagram here, the two arrows. So actually this alcoholic fermentation, right? It is of two steps. Here you have to remember, you want to be the doctor, so remember DR, decarboxylation reduction, decarboxylation reduction. So from pyruvic acid to ethanol, how will you get it? First, there is the decarboxylation. So obviously CO2 will be released, acetaldehyde will form. Then there is the reduction. Finally, ethanol or ethyl alcohol is obtained there. Am I clear? Am I clear? So it's a two-step process, one-step process. In alcoholic fermentation, your ethanol will be released and your CO2 will be released, and there the CO2 will not be released. So again, let's revise it from the handwritten PDF, right? So that, you know, we can save a time and you can get the amazing content too, because I don't want to compromise the session quality. Okay, but Chalo, so let's go. So yeah, there are very strong winds today. So let's see for how long we'll be able to take the class further. So what is fermentation? Incomplete oxidation. Am I clear? Am I clear? Done? Okay, so incomplete oxidation of glucose will occur. So, just where do you see this? You can see this fermentation in the roots of water-lodged plants, right, which are having parasitic worms like Ascaris, Tinea, right, in animal muscle. Just when we do exercises, let's suppose you are not habitual to do any exercise. Today you decided that I'll do the running. You know that next day, right, when you wake up, you have that pain that are the muscle cramps. What exactly it is? Lactic acid accumulation. We will talk about it. And there are some microorganisms as well. So they show this fermentation. So in higher plants, this fermentation is also common in seeds as well as in fruits, where the outer coverings, they are impermeable to the oxygen. There also you will see this thing. So now glycolysis, common step. So I told you it will be homofermentive or heterofermentive. Right, what will it be? It will be homofermentive or it will be heterofermentive. Homofermentive and heterofermentive. So homo may, as I said, one type of product. Two or more than two types of products will be there. So we will start with the alcoholic fermentation as we discuss. So here in front of you, you have the diagram, you have the reaction as well. Okay. And beta, in the description box, you'll find a link from where you can download the notes as well. Okay. So see, most common type occur in some fungi and higher plants, right? You know na, yeast, fungi, it is used for what? For fermentation, right? In the brewing industries, we talk about this. So two steps are there, right? In alcoholic fermentation, yes, everyone. In alcoholic fermentation, better. So in alcoholic fermentation, there will be two steps. So I told you, remember it with the help of trick. So it is DR. You want to be the doctor, so it will be DR. So D stands for decarboxylation, R stands for reduction. So it is two steps are there here. So pyruvic acid, what is the formula? 2CH3COOH, right? 2CH3COOH. So this pyruvic acid, right? Enzyme used is pyruvic acid decarboxylase, TPP, thiamine, pyrophosphate is also used there, and magnesium coion. So you will get the acetaldehyde. So there is the first step is decarboxylation. So pyruvic acid is a 3-carbon containing compound. What is this pyruvic acid? It's a 3-carbon containing compound. And what will you get from it? You'll get a 2-carbon containing compound. From this 3-carbon containing compound, what will you get? You will get 2-carbon containing compound. And here, why? Because the CO2 is released. So why are we writing 2 here? Why are we writing 2 here? Because we know that 1 glucose will give you 2 pyruvate, right? 1 glucose will give you 2 pyruvate. That is why we are writing 2 here. So now in reduction, CH3COH, this acetaldehyde. Now this NADH2, alcohol dehydrogenase is the name of the enzyme used here. So you will get 2CH2H5OH, that is ethyl alcohol, 2 NAD positive. So here you can see the energy released is around 59 kilocalories. So due to the fermentation, right, the energy released from the glucose is around 7% less energy will be released here. Is that clear? So this part is clear. Sure, beta. So next is the lactic acid fermentation. Next is what? Next is the lactic acid fermentation. So beta, it is homofermentation. Here you will see only one type of product is formed. And where will it occur? It is done in some, it is there in some bacteria and in animal muscle cells. So whenever we do the exercise, when our muscles are not toned, right, when we do such any sort of this exercise, then what happened in our muscles, right? The accumulation of lactic acid will be there. So let me explain this to you. Just say in our body there is something known as Cori's cycle. I'm talking about our own body. What happened is कि muscles में you know that muscles are having the pigment myoglobin. It's a tertiary protein. It is the pigment that store oxygen. It doesn't transport oxygen, it stores oxygen. Now, what's going to happen, students? Muscles, when they are doing some activity excessively, they demand more oxygen. They demand more energy, more oxygen. Then what's going to happen, right? When their needs are not fulfilled, so they will switch to the glycolysis only. That, like glycolysis is already there, the first step of respiration. Then that pyruvate, instead of going into the Krebs cycle, it will enter into the fermentation. It will start making the lactic acid. So lactic acid fermentation will occur here. What will occur here, beta? Lactic acid fermentation will occur here. Are you getting my point? Are you getting my point? So this pyruvate will form what? This pyruvate will form the lactate here. Now when there is the accumulation of this lactate, when there is the accumulation of lactic acid, or you can say lactate, then what will happen? We face that muscle cramps. That are the muscle cramps. Your lactic acid is accumulated there. But that what will not be done permanently. Then what will happen? So this lactate beta by our blood, it will be passed to the liver. In the liver, obviously, again, it will be converted back to the glucose. That will again form glycogen. Ya fir I would say, then glucose will be there. Then again that glucose by our blood, it will be carried back to the muscles. So this is the Cori's cycle which take place in our body. Done? Done? So occurs in homo. So this lactic acid fermentation. So it occurs in homofermentive lactic acid bacteria and muscles. So again, 2CH3COH, pyruvic acid. So 2-pyruvic acid. Again here also NADH will be oxidized. Enzyme used is lactate dehydrogenase complex. And here you have FMN, that is flavin, mononucleotide, and zinc is there. For this reaction it is required. So this is the lactic acid, C3H6O3, NAD, oxidized NAD, 36 kilocalorie. Got that? Any doubts? Any doubts? Yes, please. Any doubts? Quick. Yes, please. Clear, beta? Sure? Any doubts? Okay, just 1 minute. Done, bache? So this is the lactic acid fermentation. Okay? So now due to the accumulation of lactic acid, muscles get fatigued. So in both type of fermentation, less than 7% of energy in glucose is released. Imagine it's not 7% less than aerobic respiration. No, it is not 7% less than. Right? 7% less than. No. Less than 7% of the energy in the glucose is released, and not all of it is trapped in the form of ATP. Am I clear? Am I clear? So acid formation, even the alcohol formation, both are hazardous process beta, and this again is from NCERT. Even in microbes in human welfare chapter, I always discuss this line. Please underline it in your NCERT as well, that the yeast poison itself to death. Yeast, you know that, used in brewing industry, baking industry, Saccharomyces cerevisiae, it poison itself to death when the concentration of alcohol reaches about 13%. So that's why it basically make the undistilled beverages. Then from the undistilled beverages like beer, wine, how the distilled beverages are prepared, you know that obviously undistilled beverages are having less alcohol concentration, near about 4 to 20 or 4 to 13, 14%. Then from that undistilled beverages, distilled beverages are prepared. Do you know that distillation is done, right? Water is separated from that alcohol. So the alcohol quantity increased there, right? Just say in the case of this whiskey and all, we discuss it in microbes. There also I told you, right? Why am I mixing it? I'm just telling you that this part is also connected to that microbes wala part a bit. So I'm telling you this thing here. Okay? Okay? Right? So yeast will poison itself to death. Basically, it belongs to this chapter here, right? And I also discuss it in microbes so that you remember it. So this is the from NCERT. So yeast poison themselves to death when the concentration of alcohol reaches about 13%, right? And so it is the maximum concentration of alcohol in beverages that are naturally fermented, like your beer, wine, etc. Okay? Okay, so one molecule of glucose will yield only 2 ATP molecules, those formed in glycolysis. Where? When you are talking about the fermentation. Am I clear? When you are talking about the fermentation. Okay? Okay? Done? So this is all about it. So the next topic that we are going to discuss is Krebs cycle, or basically I would say aerobic respiration. So let's highlight it from the NCERT, then let's go forward. So the enzymes, pyruvic acid decarboxylase, important MCQ. Alcohol dehydrogenase, another MCQs, they will catalyze these fermentation, right? So bacteria, you know that produce lactic acid from pyruvic acid, right? So in animal cells like muscles, during respiration, when oxygen is inadequate for cellular respiration, pyruvic acid is formed. Is that clear? So here enzyme used is lactate dehydrogenase. Enzymes are very important, right? Enzymes are very important. So reducing agent is NADH2 here, which is reoxidized to NAD. So if I have to ask you the question, I can simply make an MCQ out of it. That during fermentation, which one is the reducing agent? Very simple question. But because we do not read NCERT or we do not practice much questions, we are not able to answer such easy questions as well. So please pay attention, okay? Okay. So next is in both lactic acid and alcohol fermentation, not much energy is released. Told you already, less than 7% of energy in glucose is released, and not all of it is trapped as ATP. Okay? Okay? Done. So what is the net ATPs that are synthesized? You know that we will only talk about the glycolysis here. So 2 ATPs. Because total 4 ATPs they formed. Out of that, 2 are used, right, already in the preparatory phase. So net gain will be 2 only. And here in your book itself, it is written, yeast poison themselves to death when the concentration of alcohol reaches about 13%. Done, beta? Done. So naturally fermented beverages will have this much percentage of the alcohol. Understood? And here, how do you think the alcoholic beverages of alcohol content greater than this concentration are obtained? So I told you this thing also, that then the distillation will be done. So that's why I gave you this explanation. So I hope up to this part all clear. Yes, everyone? Done? Sure? Chaliye, so the next part, so what? So for complete oxidation of glucose, we know that what will be there? So mitochondria and the aerobic respiration will occur. So that's what we are going to talk about. So this is the table for the anaerobic metabolism fermentation. You can see the reactions here. So trust me, these notes are amazing and they will give you everything. Okay? So let's start this process. Ready? Ready? I really like this chapter, and trust me, this is a very easy chapter, right? You just have to put your effort efforts once. Then, once you are clear with this chapter, once your basics are clear, then you are going to love it. Trust me, you're literally going to love it. Ma'am, what is the session timing? But I will finish the chapter. If there will be no power cut, I have to and I want to finish this chapter. Okay? So that next chapter we can start is ki, we will be targeting locomotion, anatomy. Okay. Done? Okay, so let's start. So let's talk about the aerobic respiration. Let's talk about what? Let's talk about the aerobic respiration. Ready? So ultimately, it is the pyruvate, right? Ultimately, these two pyruvate, they will go for the aerobic respiration. If oxygen is present, they are going to enter in the aerobic respiration. I believe this part is clear, hai na? So what is the first step that they, and where will it occur? I'll tell you. Dekho, this is my cell, right? Here I have the cytoplasm. Here, let's talk about this organelle, which is also referred to as the powerhouse of the cell. What is this organelle? This is mitochondria. Now, this pyruvate, we have to, right? This pyruvate, you have to carry it to the mitochondria because mitochondria is the site of aerobic respiration. Mitochondria is the site of aerobic respiration. We have to pass it. Now in the mitochondria, what is going to happen? Firstly, there will be a transition reaction. The very first step is going to be a transition reaction, also known as gateway reaction. Right, it is the transition reaction. 1 minute. Right, transition reaction, also known as the gateway reaction. Right, also known as the link reaction. And it is the oxidative decarboxylation. What's that? It is the oxidative decarboxylation. It is the oxidative decarboxylation. Now, ma'am, what's that? Wait. So it is the oxidative decarboxylation as well. Why this step is occurring here? Very simple. You are starting a process with, you have the 3 carbon containing compound. In this reaction, you will have there will be the formation of 2 carbon containing compound and the CO2, carbon dioxide, will be released. That's why the name of the step is oxidative decarboxylation, right? That is why the name of the step is oxidative decarboxylation. So CO2 is ultimately released, right? CO2 is ultimately released here. And what will happen? Your 3 carbon containing compound, pyruvate, it will be converted to 2 carbon containing acetyl coenzyme A along with the release of CO2. So because we have 2 pyruvates after the formation of from 1 glucose, so obviously 2 acetyl coenzyme A, 2 CO2, and it is this acetyl coenzyme A which will enter in the Krebs cycle. Are you getting my point? This is the complete step. So pyruvate will undergo aerobic respiration where mitochondria, what is happening? Very first step is there will be the link reaction. There will be the link reaction. There will be the gateway reaction. There will be the transition reaction. So what will happen here? Oxidative decarboxylation. This is actually this reaction. Your pyruvate, your 3 carbon containing pyruvate will be converted to the 2 carbon containing pyruvate. Any doubts? Yes, everyone. Any doubts? If there is any doubt, then please tell me. Any further doubts? So where that aerobic respiration is occurring, it is taking place in the mitochondria. Where is it taking place, guys? Where is it taking place? It is taking place in the mitochondria. Any doubts? Sure? Are you sure? Sure? So what will happen? So I'm writing it here. So first step clear. First step clear. So pyruvate is transferred to mitochondrial matrix with the help of special carrier proteins present in So pyruvate is transferred to mitochondrial matrix with the help of special carrier proteins present, right, with the help of special carrier proteins present in the mitochondrial membrane. And when will it happen? When sufficient O2 is available, right? When sufficient O2 is available. Understood? Guys, 1 minute.